Right, microphone's working. Well, welcome again to everybody. So I'm gonna use this one. Is it working? Yeah, great. Just give you a few words and very practical words about how to apply to UNCL and how UNCL can help you going further and going faster to the clinical applications of your nanoparticles. So I'm just going to give you a brief overview about the access procedures, the successful application profile, discuss about the lessons learned on the, let's say, 2 campaign we have performed so far, 2 selection campaigns at least, and the evolution that we are going to, and we have implemented in the call for application. So how to apply to EUNCL. As Patrick already introduced, you have everything online on the EUNCL website. There is a light application form to be downloaded, as well as an NDA to be downloaded and completed to be put into force before any application, so that everybody's protected and we have, let's say, the capacity to discuss freely of your technology, which is really, really convenient because we are really here to help you and to interact with you guys so that we understand better your technology, and we can provide you, as it was introduced by Sven Even, let's say more accurate or more relevant assays, if not in the cascade already. So we have a first step light application. It's 10 pages to be completed in the form. It's quite really easy. And then this application is reviewed internally by the core expert team of UNCL, which are the experts that will perform the assays, as well as by an external review board. Then we are, let's say, if you are selected for the second step, we are asking for a more detailed application where we will, let's say, push your material description, your existing results description a bit further, and we will interact with you either in a face-to-face meeting or in a web conferencing system, so that we really have our expert tackling your technology in order to really get the details and the substantial, let's say, details of your technology in order to really adapt our cascade to your technologies. If then again you pass this selection process, you will have the answer quite soon of being accepted for characterization, and then we enter into a really, let's say, frequent interaction in order to finalize a research plan that we will try to adapt and to be the most relevant to your technology, including all the, let's say, the assays which were early described by my colleagues earlier today. And then we will have to put, let's say, an MTA into force as well in order to define the way, the number, and the quality of the material we'll exchange. This is key as well, and this is one point I'm going to come back to later on. So the first application and the first step, which is critical, has to really focus on 4 main, let's say, criteria. First, to demonstrate efficacy in a biological system, and in vivo would be preferred, of course. Then the ability to produce 2 independent batches. It was clearly mentioned by Adriele that the material quantity, if we go to the in vivo part of the cascade, will be really, really mandatory to have a high quality and a high, let's say, quantity, a high quantity of material. Of course, to provide detailed production plan and scaling up plan, because this is going to be the further step after EUNCL, try to go to the clinics, and propose a clear strategy to transfer the technology to the clinical environment. How to provide your drug, and how to, and what would be the route of administration, for example. So this is a global overview about all the campaign, or all the TNA campaigns we have performed so far. And I'm going to describe you that TNAs are organized on a 2-yearly review period. So globally, the most frequent applicants are SMEs. We have also research institutes and universities. The technologies already, let's say, submitted are inorganic and organic ones: iron carbide, gold nanoparticles, titanium oxide, liposomes, tertimer, and virus shells as well. So we are, let's say, used to have different nanoparticles technologies. There is a high success rate, generally speaking. The applicants are proposed as really good technologies. The applications are very good. We have a lot of data. we are missing a few points, but the most critical one is the availability of the material. We have had only 2 applicants which were accepted to go into the assay cascade among the 8 which were selected for characterization, meaning that over 8 applications accepted for characterization, for the full characterization, only 2 could start it because of lack of material availability, because of poor quality of material availability. So this is really critical. We may not ask for GLP level, GLP grade material, but at least to have a quantity which is enough to go for the whole physical, chemical, and in vitro characterization at first, and endotoxin and contamination free. Of course, as it was described earlier, EUNCL is not only a characterization service, it's also a support and advice service, and the experts involved in EUNCL can, let's say, really support you in the way of preparing a better application, preparing a better batch of samples, identifying the lack, identifying the drawback in the production, things like this, or to really, let's say, help you to prepare your application better in the further steps if it has not been selected, or to support you in proposing a second step application of a higher level. And if we are in the case the application has been accepted at the second level, then we'll bring you support as well in order to set up the most relevant production process in order to gain, let's say, the quality and the quantity level requested to go through the UNCL. So these are the timelines we have so far reached. The little yellow thunder, really shows the cutoff of our selections. We are in the second TNA first session period. The deadline was April 30th, but we will push the deadline until the end of this week. So if any applications, late application or late applicant wants to apply, we really have still the end of this week to collect your application. It's really a very brief format, 10 pages. Again, very short, but very convenient, and we may, let's say, you may be able to fill in this application very, very fast. So this is, for the time being, the profile of our last application session. We have so far 7 applications, 2 more are to come. So as I just mentioned, late submission are still possible. We have more SMEs than on the previous applications, and we have different objectives. Few are cancers, other one is imaging, and another one is targeting Alzheimer's disease. So what are the main drawbacks we can encounter in the application we have received so far? As I already introduced, is the production and the material availability. So material availability, both in quantity and quality, is a major issue we have encountered. I remember that we have only 2 applications which have been concretely starting among the 8 we had, just because of this. Material sterility is an issue, and we know sterility as well, but stability is an issue as well. We need to have stable material in order to, let's say, be able to start and to finish our assays, because some of them are taking some time, and the cascade globally takes some time. So if we need material to be fully characterized, we need to have material that are going to be stable in time. The scaling up is an issue as well. We really have to have scale-up production and a scale-up plan very clearly detailed. And we need also to have production process description very clearly. So we have had some key data missing, and especially on efficacy data or stability data. How we learn? Oh, well, it was really interesting, because for the step 1, which is the light application, going to step 2 was globally easy. Success rate was about 80%. But as I just said, going from step 2 and the acceptance to the really characterization, let's say, campaign is another story, and mainly because of the material access. It's not easy, and it's really something on which I would like to really, let's say, make a point. You really have to produce and to provide with material enough and at a quality that is not a hurdle for us, meaning endotoxin free, at least. The role of EUNCL has evolved as well. We initially planned that EUNCL will be a service providing, that we still are, but we're also here to support and to provide, let's say, support and expertise to the sponsor, either to the early stage sponsor or material, or to the more, let's say, mature sponsors. And we have encountered some experience with really, let's say, large pharmas having huge issues with their product, not knowing really well the product, and they discovered things thanks to us, which we are really proud of, and we are still under discussion with them because the product and the capacities we're providing is really, let's say, high-end and, let's say, provide really good knowledge of your product, which is what the regulatory bodies are going to ask anyway. So again, we have different, let's say, maturity level of the project we have been, let's say, submitted. Some have our advanced project, like big pharma's project. Even if they don't know very well the product, they are still in clinics. They are already in clinics, sorry. And we have a really early stage where most of them are, let's say, academic projects. And some of them are really, really early stage with really, let's say, low level and low quantity of material. But we're still there to support those applications. For some of them are really promising. Globally, EUNCL has also evolved in its way of, in its application process. We are now in a continuous application process with 2 review sessions, basically April and October. We have opened to the following routes: injection and oral spray, but as well as peros. We are now open to different diseases, excluding for the time being infectious disease, because the animal models in this case are quite different. So we have to, let's say, figure out if we will be able to develop these assays for the infectious disease. And as I just mentioned, we have an Alzheimer's disease, which is quite new because most of the applications are focusing cancer. So EUNCL, as already introduced by Patrick, is involved in a really broader system, which is called the European Nanomedicine Translational Hub, and we are one of the entry point of this big service providing environment where the pilot lines should be ready to produce pilot batches. The Nanomed tab is really a strategic and, let's say, industry-oriented supports environment, and all of this is really great opportunity, free of charge. Access is free of charge for all those service providing, EUNCL as well. Just have to mention and to remind you this. We really hope to further, let's say, provide a clinical development environment, but this is a great opportunity in EU to provide you with full support from the strategic to the production and to the characterization of your nanoparticles. So here are the, let's say, main references for submission. Next review session will be October, unless you are able to provide us with an application this week. And this is the end of my talk. Thank you very much for your attention.